Okay, yeah, actually, that's actually a very important point. I forgot to mention that. All right, so we'll start again. So on behalf of the NIDDK IBD Genetics Consortium and the entire steering committee, I'd like to thank Adeeb for agreeing to speak with us. And so a little bit of background on Dr. Rahman. Went to school at Brown, did his graduate work in immunology with Larry Turk at University of Pennsylvania, did a couple of postdocs at North Carolina in cancer, and then at Mount Sinai in liver. So he has a lot of relevant experience, but he's really found his stride here at Sinai in the tech dev space. And it's because of Adeeb's work together with a very large immunology community here at Mount Sinai that we're really thriving in our capacity to do single cell sequencing through advancing new technologies. In addition to being an associate professor of genetics and genomics here at Mount Sinai, he is also the lead for the CIMAC CDC or Immuno-Oncology Biomarkers Network, which is a consortium of Mount Sinai's Pardo with a couple of other groups around the major cancer groups around the country. And so the reason why I was so happy that Adeeb agreed to give a talk to us is really his strengths in these three areas of really cutting edge. We're really at the cusp of a revolution in single cell technology development. So that when you combine all of these new technologies that immunologists have been doing on a single cell level for years, we now have these huge new capacities. This allows us to integrate genetic cells and outcomes to really define mechanisms of biology and immunology. So with that background, it gives me great pleasure, Adeeb, to introduce Adeeb Rahman to this group. So Adeeb.
All right. Thank you very much, Judy, for the intro. Screen shared. So yeah, for today's talk, just confirm, can everyone see my slides?
Yes.
All right, great. Yeah, so for today's talk, I wanted to sort of open with a little bit of sort of highlighting some of the work we've already been doing in the single cell space in the setting of IBD and just sort of the motivations for that. And then sort of transition sort of more generally to kind of technological advancements and you know, how these may be potentially integrated into sort of the new approaches going forward. And this is a minor aside, I've got multiple monitors and things. So if I kind of look like I'm staring at lots of things, just ignore me and look at the slides. So, all right. So the motivation for, let's see, there, so the general motivation for a lot of this precision medicine space stems from the fact that, you know, IBD and many other diseases, in fact, have typically been sort of historically evaluated using clinical and histological measurements, which definitely do a reasonable job in defining different disease severities and allowing us to identify different disease scores. But they obviously don't capture the true complexity of the underlying disease basis. And I think in the case of IBD in particular, one of the things that's also kind of you know, fairly notable is that there's very clearly evidence for genetic contribution to disease. I think, you know, some of the nicest examples are the familial studies that show the much higher incidence among twins and related family members. But nevertheless, those genetic loci in and of themselves have not been able to adequately explain either IBD clinical heterogeneity, adequately predict IBD disease response to treatment, or really identify clear novel druggable pathways. And some of this, I think, stems just from the sheer kind of molecular and cellular complexity underlying the disease, which I think we've come to appreciate to a greater extent as we learn more about the pathophysiology of the disease. So in addition to sort of the kind of timeline that's sort of apparent from pathological dysregulation of the mucosa, we're realizing that this is accompanied by a corresponding progressive molecular dysregulation and cellular dysregulation of different cell types and pathways. And that there's also just a lot of players involved in the system. So there's many different interacting cell types and cytokines and homing pathways. And I think a relevant point is that, you know, most of the next generation of IBD therapies are targeting these sorts of pathways. Vedolizumab, for example, targeting homing, or receptors targeting different cytokines. And so the precision medicine approach really requires a greater understanding of this complexity and leveraging that complexity as part of sort of informing drug development and potentially guiding therapeutic decisions. And so I think one example or like a vignette that I think is a nice illustration of at least the principle of this approach is the work around the IL-23 pathways. So there's been fairly strong data in mice, for example, showing that IL-22 is a downstream target of IL-23. So essentially, its expression is induced by IL-23, and that some of the mechanisms by which IL-23 may contribute to gut pathology relate to its differential impacts on different cell types, and consequently how those signaling cascades in those different cell types can contribute to IL-22 as a protective cytokine. And in this study in gastro by Bruce Sams from Sinai and colleagues, when they were doing a trial of the MEDI270 drug, which is an IL-23 antagonist antibody, one of the things that was sort of reassuring is, first, this work confirms the mouse observation that upon blockade of IL-23, there's clearly a downregulation of IL-22, so highlighting that these pathways are connected. But I think more interestingly, the fact that the baseline levels of IL-22 in the patients prior to treatment were actually able to stratify the group based on who was going to see a greater clinical response, and also who was ultimately going to experience greater clinical disease remission. So I think this sort of illustrates at least the conceptual principle that more precision monitoring of relevant disease-associated pathways are likely going to be informative in guiding some of this. And this, I think, leads to this concept that we like in many disease settings of these patient archetypes. So essentially, combinations of genetics and environmental exposures and cell types, all of which contribute to sort of distinct molecular pathways that may nevertheless be completely indistinguishable from more traditional clinical and histological sense. But by virtue of being molecularly distinct, they lend themselves to entirely distinct treatment modalities, with certain drugs being potentially tailorable to specific patients in those different archetypes. So that's sort of the kind of conceptual idea that underlines, I think, a lot of the precision medicine approaches that we and many other folks are taking at this point. And so just to illustrate how we've sort of approached kind of addressing this archetype concept in the setting of IBD, I want to highlight some recently published work from Miriam, Murad, and Judy Cho's labs, where this is really focused on performing unbiased single-cell sequencing of IBD disease lesions to try and identify or more carefully stratify sort of the underlying cellular and heterogeneity and molecular heterogeneity in these disease settings, and to see whether or not ultimately this could relate to treatment. So the approach was ultimately to collect surgical resections from advanced ileum and Crohn's disease patients, and from these surgical resections, to isolate cells from paired inflamed and uninflamed regions of the GI tract and then lamina propria, and then perform a range of assays on these. So we were doing some CITE-seq to characterize immune cell populations and their associated surface markers, single cell sequencing, and also some multiplex imaging. And so the data I want to highlight today is really going to focus on the single cell sequencing aspect of that. I also want to first acknowledge up front that obviously doing work of this sort is a very large and collaborative team-based effort. There's several folks on the research side, Miriam's lab in particular, that was leading much of this work on the computational side. Judy's group, obviously, and a large team of clinicians and surgeons for all the sample procurement. And then a lot of the ultimate assay work really hinges on the center that Judy alluded to at the start, which is the Mount Sinai Human Immune Monitoring Center, which essentially is a technology center that we've established at the institution that's intended to serve as a central hub for technology and technical expertise, and to bring that to bear in the setting of a wide range of studies to really facilitate this sort of kind of complex cellular mapping in the setting of late-stage translational and clinical research. And so in running this study, we ended up ultimately profiling single-cell sequencing using the 10x Genomics platform from 18 patients, paired inflamed and uninflamed regions, and acquiring a total of about 400,000 cells. And so broadly speaking, this allowed us to map cells into many of their sort of the large cellular compartments, and to look for differential expression or induction of these cell types in the inflamed versus the uninflamed regions. So if you're looking for what cell types are specifically found in the inflamed regions relative to the uninflamed, and there were many cell types that fell into this category, but I want to focus specifically on some of these mononuclear phagocytes, which is a key interest of Miriam Murad's lab. And so focusing in specifically on these mononuclear phagocytes and looking at them in more detail, two of the subsets in particular sort of stuck out as highlighted here in these red circles. So there was a subset of what we call mature dendritic cells, or activated dendritic cells, and also a subset of inflammatory mononuclear phagocytes that appear to be largely inflammatory macrophages. These heat maps highlight some of the very clear differential gene expression. So these were definitely molecularly distinct subsets of cells. And in looking at the genes they express, I think two of the things that were notable is in the cytokine transcripts down here, that the inflammatory macrophages make sort of a very large set of inflammatory cytokines. These activated DCs also make a set of cytokines. Interestingly, many of the cytokines being produced by those activated DCs are in cytokines we would expect to be involved in T cell recruitment, the inflammatory macrophages, and several other cascades downstream of those. And both of these cell types were specifically enriched in the inflamed regions relative to the uninflamed. And these cell types weren't present in isolation. When we consider the relative frequency of these cell types in relation to all of the other cell types that we were able to identify, we really found that they were associated with this correlated module of cells. So the presence of these cells was generally correlated with the presence of several other cells associated in the inflammatory region. And so together, this sort of constellation of cells was termed the GIMATS module. And so you can see it relates to both several subsets of activated T cells, the presence of increased IgG positive plasma cells, specific molecularly distinct stromal cell populations. And these all sort of ended up going together in this little module. But the other thing that was quite interesting was this wasn't just a general module of inflammation. When looking in these inflamed regions, it was very distinctly present in some patients, but absent in other patients. So it suggests that this was sort of a patient-specific or sort of a module that was attributed to a specific group of patients. And interestingly, when contrasting the patients here essentially with the high score versus the low score, they were entirely indistinguishable by sort of more traditional clinical or histological measures. So this sort of led to contributes this idea that the more detailed molecular mapping is able to resolve additional heterogeneity beyond what could be seen clinically. We didn't just come up with a much more complex and expensive surrogate to the CRP, like measurement, for example, which unfortunately I think is the case for many of these more complex molecular mapping enterprises. And I should note that this is an extremely small cohort of patients here that we were starting with. And so one of the things that we wanted to do was determine whether or not this sort of module was present across larger groups. And one of the ways we ended up approaching that was to mine some much larger publicly available data sets and an unpublished data set that we obtained from Janssen. So this was many more patients. And in looking, these were microarray data sets or bulk-seq data sets, I believe in the case of the Janssen cohort. But we took this sort of associated risk signature associated with the GMAT module, and in stratifying the bulk data based on this module, it was interesting to see that once again, we were able to break the overall patients down into groups who seemed to have an increased presence of the inflammatory module and those who didn't. So again, suggesting that even at a larger population level, there is heterogeneity among patients in terms of whether or not they're essentially going to be their guts are essentially going to contain this sort of inflammatory constellation of cells. And lastly, I think the other thing that was really interesting, because this just thus far just shows heterogeneity, one of the obvious questions then is that heterogeneity ultimately sort of clinically meaningful? And so here we ended up using the risk cohort, large cohort of patients who where samples were collected at baseline prior to treatment with TNF. And here, if we were to take this group of patients, and Judy can probably speak better than I can to exactly how clinical response is being defined here, but my understanding is it was long-term corticosteroid-free clinical remission during the year of follow-up following the first anti-TNF infusion. And so using that metric, if we now rank the patients based on the presence of this GMAT module, we could see that the non-responders to treatment were essentially enriched in the GMAT module, whereas those who responded were enriched in essentially the samples with low GMAT score. Again, suggesting that the presence of this inflammatory module actually has some predictive value for response to TNF therapy. And in kind of thinking a little bit about the mechanisms of what might be going on here, obviously it needs a lot more work. One of the things that was sort of interesting to consider is that a notable aspect of TNF is that it's an extremely pleiotropic cytokine on both ends. It's made by many different cells, it acts on many different cells. And generally, when considering sort of circulating TNF measurements, it's hard to tease apart that sort of cellular complexity in those relationships. But in looking at the single-cell-seq data, one of the things that we could try and do is run one of these ligand receptor analyses, where we could look for ligands being produced by certain cell types and their correlation with receptors on other cell types. So obviously, there's a lot of inference in these sorts of approaches. But what was sort of interesting is there's a lot of data here that I'm sure is not easily visible on the slide, but if you just focus on the blue arrow here, that highlights TNF. And the color of these bars represents enrichment in these patients with the GIMAT module versus enrichment in the patients without the GIMAT module. So essentially red meaning that a particular ligand receptor pair is enriched in a certain group of patients. And what was interesting is if you consider TNF being produced by T cells, there was actually no enrichment. It was found essentially across both patients. There was no enrichment of T cell produced TNF acting on ligands on either macrophages, DCs, or fibroblasts. But in contrast, if we now look at TNF production by the inflammatory macrophages, here we now did see an enrichment in the GMAT module versus not. So you can suggesting that what might be underlying sort of susceptibility here or differential response to treatment is in fact a cell type specific production of a cytokine that's now being targeted by the therapy, and leading to the idea that the different therapies may be able to, so the ability of a therapy to target a specific cell type may ultimately be a driver in its success. So that was obviously quite a small pilot study, and I think one of the main limitations, or necessarily limitation, but one of the things that I thought was a challenging study design was the fact that we were dealing with surgical resection. And so this was largely a practical consideration, because I think in getting started with these sorts of molecular mining studies, we really wanted to have a plentiful source of tissue. We wanted to be sure we had a lot to work with, and surgical resections provided a readily accessible source of that. I think the negative corollary to that, though, is that surgical resections are inevitably going to be being collected from patients at very late stages of disease. They've probably undergone many lines of therapy leading up to that point. So there's a lot of factors to sort of account for there. And so in many ways, it was actually quite surprising that we were able to see such a clear molecular signature, given all those confounders. And so currently we're in the midst of a much larger study that I think is much more specifically to be able to target these sorts of questions. It's a study funded by Boehringer Ingelheim, whereby we're essentially collecting biopsies longitudinally over the course of treatment. So we'll be able to hopefully more carefully look to see whether or not these pathways are present either pre-treatment and how they might change over the course of treatment to be able to essentially better get at this. And of course, the idea in the setting of if this all pans out and the module ends up being robust, I think using complex single cell transcriptomics is not really a tenable sort of feasible approach for a long-term clinical deployment. And so the idea is to sort of move the assay ultimately from this first discovery phase into sort of a validation phase, which we'll try and do with lower dimensional assays, see if we can apply it across these larger cohorts, and ultimately, hopefully, be able to transform it into a simpler multiplex qPCR or IHC assay that will actually allow it to be used to guide clinical decision making in the future as a long-term goal. So, all right, so I'm going to pivot there. So that was, I think, several slides to just sort of highlight, I think, the strength of these complex or precision single cell molecular approaches and their ability to resolve previously unknown biological heterogeneity. But I think, so for the remaining slides here, I want to focus on some of the kind of new technological frontiers, because single cell seq, while it was very novel when we first started this, is like a shockingly fast moving field. And there's constantly new innovations that are emerging in this space. So I'm only going to touch on a few. There's many others. We actually just held a 2-day single cell symposium that covered a whole range of new technologies, many of which I wasn't even aware of before then. So this is kind of a hard thing to keep up with. But here's a few that I think have particular added value in being able to expand beyond conventional single cell sequencing. So the first approach is a technique called CITE-seq, or combinatorial indexing of transcriptomes and epitopes by sequencing. So the concept here is using antibodies and conjugating those antibodies to a barcoded oligonucleotide. So much like doing flow cytometry and conjugating an antibody to a fluorochrome, or mass cytometry and conjugating an antibody to a metal, the concept of CITE-seq is you now take this antibody and conjugate it to an oligonucleotide. That oligonucleotide can then be used to stain cells, just as we do in those more conventional cytometric approaches, wash them, and now cells are then encapsulated using a conventional droplet-based encapsulation system or plate-based, so the 10x chromium system, for example. But consequently, by virtue of this, when cells get encapsulated in those droplets and lysed, they have essentially their native mRNA that gets released, as would normally happen in a single-cell-seq assay. But they also have these sort of pseudo-RNAs derived from the bound antibodies that also get released. And these essentially can be bound to the capture beads and sequenced in parallel. And they're, of course, linked to the cell's identity by the fact that they share the same cell-based barcode found on those capture beads. So you essentially end up with two parallel libraries, a library from the cDNA or the actual transcriptional content of the cell, a second library from the antibodies, and we can ultimately bridge the data from these two libraries by virtue of the shared cell barcode. So essentially it's simultaneous protein and transcriptional profiling. And this assay, I think, has seen faster adoption and wider dissemination than anything I've ever seen before. And so like very rapidly, it's been published from many other groups. It's been commercialized by several different entities. So I think I tend to use the term CITE-seq. It's the one that I started using I like. It was also published with REAP-seq. BioLegend now sells total-seq antibodies, which represent commercial conjugated antibodies. BD Biosciences sells ASAP-seq antibodies, which are kind of the same thing. 10x Genomics has sort of released a suite of different capture beads to be able to leverage this under the umbrella of feature barcoding. And so I'm going to be using the term CITE-seq generally for the remaining slides, but it sort of refers to this whole constellation of related approaches, which we can get into more details and questions at the end if you'd like to. And so in the early days when the technology was first released, we were definitely early adopters of this, and we did a lot of benchmarking to confirm its compatibility or the similarity of data generated with this approach versus sort of more conventional cytometric approaches, because I tend to be pretty skeptical of most new technologies when they came out. But CITE-seq was actually surprisingly reassuring in that it seemed to faithfully reproduce a lot of the antibody expression patterns that we were used to seeing by other methods. So here, for example, I'm comparing CITE-seq data up as PBMCs stained in parallel with either CITE-seq or CITE-off. And you have the CITE-seq profiles up top, the CITE-off profiles below, and the relative cell frequencies is determined by both those methods. And yeah, reassuringly, it does take some computational massaging and data transformation to make the CITE-seq data look like this, but ultimately, we can generate fairly consistent results from this as we would with approaches like CITE-off. And I think the real strength of the approach is that there's two things that are kind of intriguing. One is the idea that by using an oligonucleotide, your barcode space, so the number of possible molecular barcodes you can have for an antibody, is greater than the number of antibodies that exist in the world. So that you could technically be using tremendously large antibody panels to profile your cells. Practically speaking, as we've attempted to do that, there definitely are challenges with doing very, very large CITE-seq panels. So I think my original enthusiasm of let's profile every possible antibody has I think since been refined to let's find the most meaningful antibodies to complement single-cell-seq. But that's one kind of exciting thing. And then the other, of course, is that you do have matched antibody and transcript in the same cell. So here, for example, this is some data that's been clustered based on the antibody information. We can kind of gate this data or annotate populations just as we would in conventional cytometry to define sort of known cell types. And then we can simultaneously look at the transcriptional profiles of those same cells. So here, every column represents a cell cluster, and it's matched in either the gene expression space or the antibody space. And the critical thing is that we can share annotations across both those spaces. So we can define cells either based on gene expression or antibody or both, which provides a lot of added value. And it's also, I think, very helpful for being able to bridge sort of new genomic data sets with sort of historical cytometric data sets or sort of future cytometric data sets. And I think the other important point to note is that measuring the proteins does provide added value beyond simply measuring the transcripts in many cases. And this is a common question that comes up, especially from people who are more used to approaching sort of the field from the single-cell transcriptomic side of things. Just, we're already measuring thousands of genes. why do you want to throw in a couple of extra proteins? Like, what's the point of this? And there's several instances where we've seen significant added value, and some of it relates to just the fact that transcriptional measurements in the way they're currently done have some challenges. They suffer from technical dropout, so many times you're only measuring the most abundant transcripts in a given cell, and also the fact that not, ultimately proteins are what are mediating biology. The transcripts we're often using as a surrogate for proteins, but often that surrogacy is not as clean as we would like to be. So just to illustrate three sort of vignettes here, one kind of common example that's been well recognized in the field is in the setting of CD4 and CD8. So these are obviously proteins that are used routinely in flow cytometry and mass cytometry to define CD4 and CD8 T cells. The names of these cells are in fact derived from the expression of these proteins. But if you now look at some CITE-seq data, so this is the antibody data here on the left, you can clearly define CD4 and CD8 T cells. But if we now take these two populations that are being defined by differential expression of these two genes, and we look at the corresponding gene expression transcript of the CD4 gene, only about 4%, 5% of the CD4 T cells, which are 100% positive for the protein, express the corresponding transcript. And of those cells, most of them are only expressing, one copy, a single UMI for that. So it's a very non-robust measurement. So essentially using transcript alone makes it very hard to tease apart what's the CD4 versus the CD8 T cell. And there's probably some biology underlying this. So I think the CD4 gene is one that tends to not be regulated in such a way that there's a large pool of CD4 transcripts floating around. But nevertheless, it leads to potential confounding factors in interpreting your data. Another example is a CD45RA. So the CD45RA and CD2L, these are again two proteins that are routinely used to define sort of naive versus memory T cells. But CD45RA is a splice variant of the PTPRC gene. And so while there are some computational tricks you can go to define splice variants, generally speaking, looking at expression of just the regular gene is not going to adequately allow you to capture sort of these sorts of changes. And the third example is this antigen called CD57. So this is an antigen that's expressed on late-stage terminally differentiated NK cells and CD8 T cells, CD4 T cells. So it's a very useful marker in flow cytometry for defining sort of T cell maturation and exhaustion. However, the CD57 epitope turns out to actually be a glycoprotein modification of another protein. And so since it's post-translational, again, it's kind of hard to predict from the transcriptional level. One of the things we can try and do is we can look at the expression of the enzyme that actually mediates that glycosylation reaction, B2GAT1, to see if that can reproduce this, but clearly it doesn't. So essentially the enzyme itself can't predict the expression of CD57. So there definitely are certain protein markers that are hard to predict directly from transcript. And this is where I think CITE-seq really provides the benefit of being able to add some of these to a panel. And sort of repeating this exercise, across larger panels of proteins. So this is about 20 or 30 antibodies where we've done a similar sort of exercise. In each case, we've taken an antibody, we've defined a cell type as being 100% positive for the expression of that antibody. And then we've looked to see what's the corresponding expression of the transcript in those cells. And you can see there's a lot of heterogeneity. So some genes, like way down at the bottom, if you look at CD161, for example, a marker of mucosal associated invariant T cells, almost 100% of the cells that are defined as CD161 in the protein space express the corresponding KLRB1 transcript. And so this is very faithfully reproduced, and it's one of the reasons if you look in single cell transcriptional data sets of T cells, they almost always will annotate mate cells, which again is a somewhat obscure cell type in traditional flow cytometry because most people don't include the markers to identify them, but nevertheless they stand out very clearly in the transcriptional space. But now if you look at kind of the opposite end of the spectrum, something like CCR5, chemokine receptor, also found on mate cells, is almost not expressed at all in the gene expression space. And so, yeah, again, I think this provides kind of a framework for thinking about what are antibodies that can provide significant added value beyond what you find by transcript alone. And sort of in new experiments that we're running in Judy's lab, there's several sort of specific IBD-associated proteins where we want to evaluate this sort of pattern and see whether or not adding them into a stain will allow us to define things more carefully. There's also some other kind of interesting things that end up emerging from CITE-seq. So one of the other sort of dirty secrets, I don't know if secret is the right term because it's probably quite widely known at this point, but one of the other things that is known in the single cell seq field is that we really don't capture granulocytes. So One of the things we've noticed just in doing kind of comparative CITE-off versus traditional single-cell seq analyses is the one population that was always underrepresented or almost never represented in single-cell seq were neutrophils and eosinophils. And CITE-seq has actually kind of given us some more insight into what's actually going on there, because this is a whole blood sample where we've defined the cell types based on antibodies, and we can readily see neutrophils here in orange and eosinophils in blue. But if we now look at the corresponding gene expression transcripts on these cells, you can see the neutrophils that have almost no gene expression, and similarly with the eosinophils. And so here, this is just looking at the number of the overall UMI sum and the unique genes across these different cell types as defined in antibody space. And it's really highlighting that these cells are just barely have any transcript. And so if we simply, as most people do, take a traditional single-cell seq data set and we threshold to identify cells based on the presence of gene expression, we essentially miss these cell types. And so while CITE-seq doesn't allow us to perform transcriptional measurements of them, it does allow us to at least account for their presence more effectively in samples. There's also other sort of practical considerations that are sort of improved by CITE-seq. So what I've been mentioning so far is using antibodies against specific surface antigens to define specific cell types. Another flavor of CITE-seq, which is something that we've been doing in the CITE-off world for a long time, turned into barcode in the CITE-off world, it's been coined as hashing in the CITE-seq world, is to use antibodies against ubiquitous surface antigens to define all the cells from a given sample, such that those cells can then be combined together and pooled and analyzed as a single sample. And so this provides sort of added practical benefits, one from cost, because you can kind of leverage more samples in a single lane of a 10x chip, but also from a data quality perspective. So one of the things that I think relevant things in the context of IBD, for example, is to collect multiple biopsies, for example, from geographically distinct sites, hash them, pool them, and analyze them as one pool, or potentially collecting pre-treatment and post-treatment biopsies, cryopreserving them, and then pooling them for kind of a combined analysis. So I think this really allows us to sort of reduce technical variation associated with independent sample processing and kind of improve our ability to resolve maybe more subtle signals and differences between samples. And lastly, I think CITE-seq also offers a pretty cool opportunity to be able to really interrogate sort of therapeutic responses in ways that really aren't possible otherwise. So this is an example from some of our work in hepatocellular carcinoma, where we have patients on a BMS trial who are being treated with nivolumab, so an anti-PD-1 antibody. And so one of the consequences of getting an anti-PD-1 antibody is it essentially blocks the conventional PD-1 epitope that we would use for detection of PD-1. But we can actually detect the bound antibody by using an antibody against the IgG4 isotype, which is pretty rare in normal circulation, but is the isotype of nivo. And so essentially we conjugated an anti-IgG4 antibody for CITE-seq. And now here this essentially is a resection sample from a patient who received treatment. And you can see that looking in the tumor, this is looking at the CITE-seq data, we can identify our CD8 T cells, and we can actually detect the bound of nivolumab on a subset of those cells in the tumor, but not in the adjacent normal lung. And we can now look at the gene expression of these cells, and we can see that the cells that are bound by nivolumab have a distinct gene expression signature. And so it allows us to actually look at the specific transcriptional changes that might be being induced by the presence of a drug or an antibody, which I think is another very powerful approach that can be taken in some of these studies. All right, so that's CITE-seq. So it's an approach that, again, I think was extremely novel very recently and is somehow not novel anymore because this field moves in sort of an insane and crazy way. But there's even more innovations that continue to emerge. So just last month, the New York Genome Center Technology Lab, the same lab that sort of initially published CITE-seq, has just released a technique they're terming ASAP-seq. So ASAP-seq essentially is to ATAC-seq what CITE-seq is to single-cell seq. So essentially it allows you to couple single-cell ATAC-seq measurements also with antibody profiling measurements, which I think also has a lot of interesting added value. Admittedly, the ATAC-seq space isn't one that we've done too much work in, but obviously looking at epigenetic changes is very powerful. Being able to then link that to kind of historical cell identities and all of the kind of constellation of things that I've mentioned are the advantages of CITE-seq offers a lot of added value there. So being able to both couple cell identity with the sort of epigenetic modifications. And then sort of a third major frontier is multiplex spatial imaging. So I think all of the work I've shown today has really been from dissociated samples And I think that's useful for identifying cell types and molecular pathways in those cell types. But obviously, the immune cell and the immune system in particular is really defined by cell-cell interactions. Like most of what immune cells are doing are communicating with other cells. And so physical proximity in tissue is a really important metric to be able to gauge what's happening in some of these contexts. And yeah, the approaches to do this are kind of getting progressively more interesting. So just one example here is going back to that original story I presented on this inflammatory EC and inflammatory macrophage story. If we take a gut biopsy and we stain it with sort of CD68, which is a marker for pan macrophages, and DC lamp, you can see already it's kind of interesting that the macrophages are distributed pretty uniformly throughout the tissue, the little pink spots here, whereas the dendritic cells really seem to have much more sort of localized expression patterns. And if zooming into this area, we can see that DCs are often present in lymphocyte aggregates with a lot of T cells and B cells, which is sort of consistent. You might recall kind of very early on when I showed you the transcriptional pathways being induced in these cells, the DCs seem to be expressing many cytokines and chemokines associated with T cell recruitment. And so it makes sense that that might be what's going on there. And so in considering the role of different drugs, especially things that are expected to influence T cell or cell homing, things like vedolizumab, for example, looking at the ability of these drugs to impact the spatial rearrangement of cells and tissue, I think, is a very powerful approach. And this is a very simplistic multiplex immunohistochemistry approach that we've used for a few years now. But this is another space that is incredibly busy, and there's an ever-expanding range of technologies to target this. So, for example, there's mass spec-based approaches such as Hyperion imaging from Fluidigm. That's what this data is over here that I'm showing you. There's also multiplex ion beam imaging from IonPath. So at Sinai, we made significant institutional investments to bring in one of the beta instruments there. So that's another machine we've been working with. It's been challenging, but obviously has a lot of potential. There's the Codex system from Akoya, Maxima from Miltenyi, Orion from Verisight. I mean, the list just keeps going on. I get emails probably once a month from a new company that's just come out with some new approach that they want us to evaluate. And then even more so, like another even more rapidly emerging area and kind of progressively proliferating area is the whole frontier of spatial transcriptomics, rather than simply looking at cell types based on antibody, defining cell types in situ in tissues based on transcription. So one of the kind of, I think, most widely used, or certainly one that's being quite widely adopted by the community, is the 10x Genomics Visium platform, partly just based on its ease of use. So this is a platform that allows kind of unbiased 3' capture with spatial context. One of the limitations is that the resolution isn't that great, so it's not single cell resolution, it's more sort of regional resolution, but still nevertheless a pretty powerful technique. And then there's techniques like probe-based approaches, things such as MERFISH, which allow sort of they're essentially probe-based detection, but they have exceptional resolution, allow detection of lots of different targets, but are sort of incredibly difficult to establish in the lab, which is, I think, one of the reasons that they've had sort of poor adoption by the community. But certainly I would anticipate that in the coming years, those are gonna only become more accessible and more powerful as time goes on. All right, so that's what I have essentially for, I wanted to leave a reasonable amount of time for discussion and conclusion. So just to conclude, opportunity that our single cell mapping work really identified the cellular pathogenic modules that are present in diagnosis and that seem to be able to correlate with resistance to anti-TNF therapy. This kind of allows us to go beyond IBD genomics alone and really start to consider disease pathophysiology together with things like diet, microenvironment, cellular organization of the gut tissue, and all these factors contribute to the lesion. And ultimately, this sort of deep understanding of that cellular molecular heterogeneity is hopefully going to be informative in defining both mechanisms of disease and mechanisms of treatment resistance and identifying novel therapeutic targets. Yeah, and essentially that this is, there's an ever increasing range of tools and technologies to extract ever more information. And I think a real challenge is often how best to define the most appropriate tool to address a particular challenge, and of course, to be able to adequately benchmark these tools and apply them in their most appropriate context. And I just want to end again by acknowledging the very large group of people who've all contributed to this work and this kind of our ongoing initiatives in the space.
Thank you so much, Adeeb, for just a wonderful talk. We have a question from Ken Lau. Colleen, so if you can unmute Ken.
Hey, Adeeb, how are you?
Good, thanks.
Nice to see you.
It's been several years.
Yeah, so hey, I just have a quick question. So you mentioned about the lack of detection of neutrophils from the 10x data.
Yep.
I'm just wondering, from what you understand about this field, is that specific to the 10x platform, or is that specific to all single-cell RNA-seq platforms?
That's an excellent question, and we literally have some data coming off the sequencer this week that will hopefully give us some experimental information on that. I do think it is. I think the 10x platform is particularly susceptible to it. So anecdotally, I've seen some in-drop data where I've seen at least some preservation of neutrophils, and then there's also been some data in the setting of some of the COVID work. There's been a few studies that have published the presence of transcriptional signatures of these low-density neutrophils in COVID patients using the Rhapsody system. So we're actually doing an explicit benchmark to compare samples with no neutrophils across a few different platforms to see if this is a 10x specific phenomenon. So. And I should maybe note that we've poked into this quite a bit. It was kind of tangential to this talk, but it seems to really be due to, it's not because neutrophils don't have RNA. Right? That doesn't make sense. Like neutrophils clearly have RNA. We can pick them up in bulk C. It seems to really be that they have a lot of RNases that in fact degrade their RNA. So upon getting encapsulated, it seems like whatever the RNase inhibitor cocktail is present in the 10x droplets is insufficient to inactivate those RNases. So we have some pretty strong data showing that's the case. But despite sort of pumping more RNases into the system, RNase inhibitors into the system, sorry, we haven't been able to recover it. So.
Okay, we have a question from Madhu that actually maybe I should kind of answer about what's the significance of the geometric mean on GIMATS module, and is the module available publicly? So all of the data that Adeeb presented is on Geo, so it's fully shared. So all of that should be shared. We noticed a little gap in our sharing and that we didn't share the PBMC data. So now hopefully all of it is shared. And then in terms of the significance of the geometric mean, I think we kind of use that for when you were trying to compare an equally weight kind of distributions of cell types that are substantially different. And so it was just a kind of a practicality purpose for the use of the geometric mean. I don't know, Adeeb, if you want to weigh in on that, but
I agree with that.
Okay, so any other questions? Quick question about what you think in terms of kind of a phenomenon in Crohn's that I think has been described quite a number of times, which is the in situ mature dendritic cells. And put your immunology hat on in terms of like, what do you think is, what's the significance of that? Because that clearly is a major component. Mm-hmm. And one of the chemokines that the mature dendritic cells produce is CXCL9, which we're seeing as a big biomarker for treatment non-response. So, if you're immunologic.
Yeah, no, I agree. I mean, it definitely seems to be an implicated cell type, obviously one that we're very interested, Miriam's lab is certainly very interested in. I think one of the things that is interesting, as we've been doing a lot of the single cell sequencing in the setting of multiple different studies, in the setting of cancer, Miriam's group has recently published a paper on a cell type that they're now terming mReg DCs for mature regulatory DCs, which we find in tumors. And in characterization of these cells, I think the defining attribute is really that they are DCs that have consumed antigen. So that's what's driving their maturation. And so it's a specific antigen exposed DC, and as a consequence of having consumed that antigen, they're upregulating this kind of homing module or this module of a range of chemokines that end up recruiting other cells into the space. So this certainly doesn't seem to be an IBD specific phenomenon, and so hopefully there'll be some insights that end up kind of getting generated in some of these other fields in terms of targeting the cells that might be informative. But yeah, I mean, we definitely think of DCs as being a cell that's very early in the pathway of activating an immune cascade. So the suggestion that the same thing might be happening here, I think, is very valid.
Any other questions?
There's one question in the chat from Thomas asking, does GIMATS module correlate to the presence of TLS in tissue, similar in makeup of cells? Could this drive resistance to anti-TNF?
I'm going to punt that one to you, Adeeb.
Let's see. Does GIMATS model cause the presence of TLS in tissues, cells? Could this drive? Oh, I see. You know what? I don't know the exact answer, but I would imagine it does, just based on the nature of the cells that contribute to the GMAT module, including the plasmablasts, for example, Ig positive plasmablasts, which are typically found in some of these TLS structures, would suggest to me that they likely are correlated. But I don't know if we've explicitly looked for the correlation. But I would think yes, probably. Could this drive resistance to anti-TNF? So I guess is a suggestion then that could the presence of the TLS in the tissue ultimately be one of the factors driving resistance to anti-TNF? Which again, potentially would be a yes. And this interesting, I always tend to be a little, I mean, despite their power, I always tend to be a little bit skeptical of some of these inferred ligand receptor kind of inference networks, just because there are a lot of things that can contribute to that. But I think the interesting suggestion there is that if T cells in general, looking at sort of the overall T cells isolated from one of these samples, the T cell TNF by itself doesn't seem to be related to the presence of the module. It could nevertheless be possible that you have a small sort of subpopulation of T cells that are TLS derived that might have different productions there. So again, that's where I think some of the spatial approaches of being able to profile cells in the context of, let's say, a TLS versus ones that are more disseminated to the tissue could be very informative in getting at questions of that sort.
Can you update on like where you might be in terms of single cell ATAC-seq in your thoughts?
We are very early, so I would say we're just getting started in that space. I mean, I think several other groups have done a lot more work in that space. It was something that there was so much we could do with single cell transcriptomics that it wasn't something we embarked too much on. But I will say that, I mean, one of the things that I think has really driven kind of the expansion of this field really has been 10x genomics. I mean, these kits that they provide really take these technologies that were quite inaccessible and hard to implement of your own accord, and they've made them just very readily accessible. So I mean, the fact that you can just buy a kit and with relatively minimal expertise get ATAC-seq data, I think it's fairly powerful. The ATAC-seq data is, unfortunately, it suffers from an even greater, like the issues of sort of data sparsity and dropout and things like that are an even greater problem there than they likely are with single cell transcript, because you literally only have 2 chances to pick up what you're looking for. So I do think the computational methods required to analyze ATAC-seq data, again, require a lot more inference of these sort of networks. So I think one of the interesting considerations is a lot of these sort of supposed single cell approaches really aren't single cell, because essentially the information from any individual single cell tends to be largely uninformative. It's really about being able to group cells into related categories. And so it's maybe provide sort of heterogeneity that can help guide more resolution of the tissue. And I think that's particularly the case in ATAC-seq, single cell ATAC-seq.
Any other questions from the group? Well, in that case, I'd like to thank Adeeb for a truly outstanding presentation. Thanks, Adeeb.
Great. Thank you all.
So, yeah, great job. I never know whether you should wait for the applause afterwards in the Zoom era.
Yes, I'm judging everyone.
Never to applaud, people are muted.
Disheartening, yeah.
Thanks again. Well, yeah. Terrific job.
Take care, everyone. Bye.
Goodbye.